genomes. And if we had to line to. And then we've also created a reference graph. So we did long-read sequencing, we did hi-fi sequencing for about 25 genomes, and we've created an actual pan-genome reference graph on that. And then we've also been collaborating with Professor Lamarca to do telomere to telomere genome. So this means like the complete genome. So the normal human genome is actually not complete. It's missing bits in it that are really hard to sequence. Then you can have these new techniques, which is different kinds of long-read data, which allow you to resolve those regions, and you can literally get telomere to telomere, every piece of the human genome. And so that's again, these again have only been done for European populations. And so what we've got is some samples from the DRC, where we've got 3 types of ONT long-read data, and we're just finishing hi-fi data as well. And so we're about to, we'll be basically assembling these into telomere quality genomes. And this will allow us now to have a much better, complete reference genome from Central Africa that can be used for aligning genomes. And the HPLC did a lot of capacity development there. They came, we got a P24 machine in to do this, and they came and did wet lab training for this. We've also got an opportunity to work on other African long-read data. So they're taking the 1,000 genome samples, and they're doing 2 things. So some, they have the sample themselves, and labs around the world can apply to get the samples, do all the sequencing and everything. And then for some of them, they've actually generated the data already. So one of my PhD students is taking a few of the samples from Africa that has all this beautiful long-read data, and is doing the actual assemblies for the International Pan-Genome Consortium to get these telomere to telomere genomes. So it's a way for the Africans to actually generate these T to Ts for our own populations. So what is these T to Ts? And just one quick analysis. Somebody took a patient sample. We had people 2 different kinds of graft genomes. And what they found was that you get more variants called of the linear, in this case of the linear than the graft, but because it's accounted for natural variation, so you're actually removing what is actually not a variant. But there was a lot more novel variants in this graft than the linear one, and these novel variants actually had some clinical implications. So when you run this through, they actually through variant annotation, they actually have potential pathogenic features on them. So it's really important to have the right reference genome, and more people are starting to, the tooling starting to come about for people to actually use reference graphs and TTTs and to improve this. So you could build it for your population if you always know you're going to be working on it in a particular country. So just to summarize the resources developed by H3Bionet in terms of this genomic data ecosystem, from the data collection, from sample collection, these standard case report forms. From the data generation, we actually designed a new chip, and then another version is now being designed on a smaller chip. We've got all these data analysis workflows and tools, including the pan-genome. And then we've also got some downstream post-GWAS analysis tools and visualization tools. And then we provide an archive for you to submit that data to public repositories. And then I'll talk a bit more about access to the data. So we now have, we still have an extreme scarcity of African data, but there is some out there. These are some analyses done on what we think is out there. We also are working on a project to sequence 25,000 African genomes, and then there's some other projects, the Agenda Project, and some other upcoming projects. GenImpact's just been announced. It's a welcome-funded project to increase the fill the gaps in diversity in African data. And the problem is that even with this, the data increase is slow. So yes, we are increasing, but the rest of the world is increasing at a much higher pace. So that gap between the amount of data that we have versus what everybody else has is just increasing. And the problem is that even when we do have the data, it's not necessarily findable. So when we created the map on the right, it actually took a while to find these data sets, and they're not always findable. It's not always accessible. You have to like email somebody, do you have this data set still? And that moved on. So it's really, really hard to actually find the data. So what we've done in East Africa is to make the East Africa data sets findable is we have a data biospecimen catalog, so you can come in and find out about the East Africa projects. You can search for the data, it tells you which data sets there are, how many participants, whether those samples are also in the East Africa biorepositories. And then once you've kind of searched for them, depending on your criteria, I want females from Nigeria that have diabetes, then you can actually submit an access request to the Data and Biospecimen Access Committee. If your data set's not East Africa, in a new project, we've actually got Alwazi's an open data science platform that I'll mention a bit later. We actually have a catalog in here where this is now not just genomics data sets, it's health data sets, demographic data sets, climate change, all sorts of different kinds of omics data. And so you can actually search this, and then the get button will tell you how to go about accessing this data set. So how we get the data in here is that if you have your own metadata for your sample collection or your cohort, and you've got a code book, you can actually input that. A large language model would kind of will map that, and then it gives you this output where it says, okay, these are all your variables, is this what you meant? So it maps it to kind of a common data model. That's all done in a RedCap form, and then that can be automatically scoped into the Alwazi catalog. And so the catalog has information about the projects, and then which data sets they have. And then, as I say, it's all different types of data sets, and all coming from many different countries. So we have, these are some of the countries where we have data sets from, and the kinds of data sets. So it's a way to actually find the data sets if you want to be, comparing your data. Say you want to create your own graph genome or reference genome. How do you actually go about finding a data set for your country? So once we found them, how easy are these data sets to actually use? So you want to so you've got your genomic data. It's just huge. You want to actually be able to interpret this data. You don't want to necessarily be able to have to download FOSQ files or the CRAMs, or maybe you just want to work with the VCFs, but now you've got individual VCFs for 1,000 people. Do you want the joint call? So you need to think about what part of the data are you going to actually be using? Or do you want to actually use a transformed version of the data where you don't care about those all these file formats, you just want a nice web interface where you can access the data. So if you want to find SNP level information about a particular, for your patient, you can search, say it's a pharmacogenomic variant, you can search pharmGKB, for example, and it gives you information about that. But it doesn't say who that information is relevant to. It just says, okay, this variant has been known to affect response to this particular drug. You can then go to Nomad and look at the frequency of this variant. But again, so it breaks it down a bit here. So it says African, American, European. Africa's not just to say what the frequency is in African populations. You can see here, in fact, that the frequency of this particular variant is reasonably high in Africans compared to everywhere else. And then, so you can go and find more information about this. But what if you're pulled out all the African relevant information and actually read the publications and trying to say which population was the study actually done on. So when you look at that same variant, you can actually see that there was a study done there were two studies done in Central Africa, And you can go and see, well, how relevant was the SNP to that particular population, because I can go and look at that actual study, as opposed to just getting the generic information for everyone in the world. Same with the aggregate data. So we created the African Genome Variation Database to look at more fine-grained at African populations. So that variant, if you look at that variant across different regions in Africa, you can see that in Central Africa, the frequency is quite low, even though the overall frequency in African populations was high. But there's huge variation across different African populations. So this is very relevant now if you're working on a particular population in one of those regions. So this is how we've tried to make the data a bit more useful. We still link to all of these other databases like Nomad, ClinVar, ClinGen, etc. But we just tried to make it more granular so that it's more relevant to African populations. If you now want to think about working with a larger data set, so that's looking at one SNP at a time, you now have to think about, I've got big data sets, how am I going to manage the data, where am I going to compute, where are the data sitting, how am I going to integrate that with the health data, etc. And there's also challenges of moving data across borders now, because you've got big data sets. It's not just summary data anymore. So the Open Data Science platform that we have is basically a platform that allows you to find data, for example, in the catalog, and then access the data if you can get, you follow the link to how you request access to the data. When you've got your tools, so those workflows I was talking about, do you want to do a GWAS analysis or just NGS variant calling? Those tools are available in something called DocStore, so it's an online cloud accessible environment. And then you can actually run the analysis in whatever environment that you want, whether it's a cloud environment or on your own HPC. And then we have a web interface where you can actually run these, do this fetching of data, running the analysis, and then sharing the results. But we've also come across, even with that, the Awazi platform right now will work on a specific computing environment. So your data, if you're going to compute on AWS, your data must be on AWS. If you're going to compute on your own HPC, your data must be there. But sometimes the data sets are all over the place. We know that after the African Union introduced their data policy framework, many different countries have implemented these data protection laws. Data can't necessarily move. In many cases it can if you have equal protection in different countries, but there's starting to be barriers to move data. And also when you're working with health data, particularly electronic health records, the Department of Health is not going to want to move that data across borders. So as well as having a nice environment to be able to compute on, in a single spot, we also need to think about how do we manage analysis of data sets when they're sitting in different places. So what we've done is we've actually implemented a pilot project. In this case, we did joint variant calling, where we divided 1,000 genome data set and put one part of that data set in Mali, one in Uganda. We basically created, using GA4GH standards, a way to be able to send your analysis to each of those places and get the results and combine them. So you're not taking the data, you're literally visiting the data and letting the results come back to you to analyze. So this is a whole new thing that's coming up now, is this federated analysis, federated trusted research environments, so that you can start to protect your data, but still make it accessible, because accessibility of data is extremely important when you're trying to think about the health of your patient. The information you get from on your patient can very much be informed by global data. If you can't see or access that global data, you're at a distinct disadvantage, and you might not get the right information. So it's important that we be able to facilitate this federated analysis. And we're working with a company called DNA Stack, who also part of GA4GH in Canada, where we have a nice web interface on top of this. So you can basically go and you can look at these different data sets, and you can say, right, I want to, if you've got access to the data set, you can go in and say, right, for these patients from this country, I've got these phenotypes, I'm going to pull that data out, run an analysis, so I can run a GWAS, or I can just look for, have you seen this variant in a particular case with the following phenotypes? So there's still a number of gaps. So African genomic data are scarce, but the linking of African genomic to phenotype data is even more scarce. It's very difficult to find data sets that have genotype-phenotype linking. So we're trying to work on that. We're using AI tools, mining the literature, mining databases to get more relevant to our populations. And that might differ whether you're in North Africa or South Africa. Clean, harmonized clinical data. very difficult to get because it's very sensitive. So we really need to improve this kind of link between the health and the genomics. We need strengthened health systems to do this. We need government buy-in to do this. The governments need to recognize the importance of getting clean health data and links and generating genomic data. And then we need this federated infrastructure. We need to catch up with the North, because the North are routinely using genomics for health. They're routinely doing genomic screening, newborn screening. They're using polygenic risk scores. They're actually doing disease prevention now because they have this information, and we need to catch up. In order to do this, we also need a massive genomics workforce. So in Urbana, we had kind of this multimodal way of building a workforce, training trainers, training the users, training scientists through a mixture of online courses, face-to-face courses, hackathons, internships, trainer, all these different modes of actually building your genomics workforce. Sometimes it works just to do online training if it's theoretical. Then you bring people together to analyze data. So we bring your own data, hackathons, jamborees. We do this as a way to cement the knowledge and get to actual results. So we also designed this to be very much dependent on who the audience is. So if you're a geneticist or clinician, you don't really want to know the nuts and bolts under the hood of what this workflow is doing, what the algorithm's doing. You want to know how to run it and how to interpret the results. If you're the one creating those tools in the first place, you need to know the nuts and bolts. So we had to design the genomics workforce training plan depending on who the audience is, and have it, from applied to technical. So we had different courses for these different groups. And so when you're thinking about increasing capacity with genomic medicine in your own institution, think about who you're targeting and what they need to know. Look at their job description and use that as a potential for what is the competency that they require, and then train them accordingly. We also have been working in pathogen surveillance and public health. So a lot of the training we did previously was in academia and building skills in researchers and clinicians, in students. But when you train in public health, you've now got a workforce that's very busy. They have a full-time job. So the kind of training you have, it's hard for them to take 5 days out to do training. Maybe you have to do it in the evenings. Maybe you have to do asynchronous training, etc. They have immediate needs. So when we started this program, the pandemic started 2 months later. So immediately we were training on SARS-CoV-2 analysis. Then monkeypox came along. Immediately we had to create a curriculum for monkeypox and train people in that. So it's very responsive. You have to be very responsive in this kind of training. Whereas in the genomic medicine side, there are a lot of new things that happen, but they don't happen at a pace that you have to quickly run a course and get these up and running. So it's a different environment, but sometimes when you're in a clinical kind of health setting, you need to also think about you training nurses. They're busy. They have a full-time job. When can you train them, and how do you give them just the knowledge that they need to be able to do their job or implement something new in genomic medicine? So we also found when we were doing this that there was a need for standards and training, and this is something we did for pathogen surveillance. Was we actually got the entire community together and developed a standard curriculum, because Africa CDC wanted a standard curriculum in different areas, aspects of pathogen surveillance. So the reason was because somebody would, so during the monkeypox outbreak or the SARS-CoV-2 outbreak, somebody would come to them and say, I can train on SARS-CoV-2, I can train on, and different people were coming, and when they were training in different regions, they had no idea whether the participants in those regions were getting the same level of training, because there was no standard curriculum for people to actually implement. So we designed this curriculum so that it can actually be rolled out anywhere, and it tells you at what level. So it's either the introductory, intermediate, or advanced. We've got these generic courses, which are, what is genetic epidemiology? What is NGS? What are the ethical considerations about this? What is the general sort of study design? And then very specific ones in terms of pathogen-specific courses. So if you're working on SARS-CoV-2 or if you're working on cholera. And so this is what a module looks like. So it goes into detail about the depth at which you teach it, number of contact hours, what are the learning outcomes, and then the indicative content. So I think this is something that maybe, through a course like this, we could probably develop in the genomic medicine space, so that when people are training in genomic medicine, they're learning something similar. I mean, obviously you'll have your own use cases depending on where your context information. So to conclude, before I go on to the kind of career development stuff. When you're working on medium to large scale genomics projects, you really need to think about the data ecosystem. So you need to think about, you're now dealing with very sensitive data. You can't just email it to somebody. You have to think about encrypting it. G4GH has this Crypt4GH tool. We can help with encryption. Your local IT department should be able to help with encryption. Think about where you're going to store the data. If somebody's sending you data that's encrypted and it's 10 terabytes, you need 20 terabytes of storage because you've got to get the encrypted parts, and then you need the space for the unencrypted, when it's unencrypted, and then you can, remove the rest. Little things like that is really hard to think about if you don't know about these things when you're trying to design your own genomic data ecosystem. How do you transfer the data? We use Globus Online because in Africa, the internet goes down. Globus Online can pick up where it left off, where some FTP protocols can't do that. So we've spent literally more than a decade trying to work out what are the best pieces of the genomic data ecosystem that work for our settings. And it's not a plug and play thing, so you've got to get each component sort of separately, but I think we have enough experience to help you if you need to set this up from scratch. What kind of HPC do you need? So, you've got your normal processes. If you're creating a graph genome, you need high memory machines. If you're just doing alignment and variant calling, you don't. So how do you set up your infrastructure to manage that? If you're doing AI, you need GPUs. So you need to think about what is your endpoint when you're setting up your genomics data ecosystem. And then the governance, of course, is extremely important. Who can access the data? Once you've said somebody can access the data, how do they physically do that? So in G4GH, they've got this what they call passports and visas. So the passport is saying, I know who you are. It tells me who you are. You're a bona fide person, researcher that is recognized. Then the visa says, you have a visa for this data set, which means that you have been authorized to access this specific data set. And it's a whole technical implementation so that when you've got your genomic data infrastructure and you've got people trying to log in, these tools will say, this person is allowed to log in and see this data set, this person isn't. So it's a lot of kind of technical stuff, but it's important if you are going to be setting this up from scratch. But even if you are trying to access the data set, how do you physically do that? There's an increasing need for federated analysis. So think about whether you are going to have to access data from somewhere else. In many cases, you're just going to analyze your own data. But I think that you are doing yourself a disservice if you only do that, because there's so much data out there that's going to actually inform your own data. It puts your data into context. It puts your population information into context. You may think a variant is really important because now it's low frequency in your population, it's been linked to the phenotype, but in another population nearby, maybe it's actually quite common. So context information is very important. And it's important to build a human capacity for data management and analysis. And the other thing to consider is not just thinking about the small picture. Think bigger. So your health is not just about the genetics and the environment. It's about everything you interact with. So there's the new One Health approach, which is now looking at everything, even your microbiome. So genomics is not just about your genome and your phenotype. Start with that, for sure, but also then think about the rest. There's a lot of things that impact that, and that can give you the big picture. So that's everything on the genomic data infrastructure and ecosystems. Just now a little bit, I was asked to talk about consortium and network building. I think I have had the biggest network and consortium in the university because our contracts office was always fighting with 32 institutions every year to do contracting. So I have a bit of experience in building and working with networks. I also had a consortium when I was in the UK working at the EBI before I came here. So I'm going to talk a little bit about building a network, the management of those recommendations and sustaining a network. So I think the most important thing is to make sure that you build equitable partnerships and networks. So first of all, find partners that we all want to work with our friends, that's great, but do they have the right expertise for the project? Sometimes it's nice to bring people along because you know that they deliver, but they still need to have the right expertise for the network or the overall goals of the network. It's important in these partnerships to respect all partners and their contributions. So it's really easy to just, this person's not doing anything. Maybe there's a reason they're not doing something. Maybe they're coming from an environment where they just don't have the capacity to do something. So just respect, and when they bring up an idea, listen to that idea. Don't dismiss ideas. Just think about everybody. If you were in that situation, would you want to be dismissed? And make sure that everybody has an equitable role, especially when we're working with foreign collaborators. It's often I often see that kind of the collaborators in the north have the upper hand on these collaborations, and we're just the sample collectors. Make sure you are up there. You have an equitable role in the partnership. So to build a successful network, you really need to know what are the aims and deliverables of the network. You don't just build a network just for the sake of it. You're doing it because you're trying to achieve something. And each person must have a particular role in that deliverable or that set of aims that you're trying to achieve. And so each partner should be delivering on that, and you should be able to monitor that. But don't just put people down, as I said, if they're not contributing. It could be that there's a reason. In many cases, in my experience, there's sometimes just not a reason. They just don't do anything. Some people talk and don't do. And then you definitely get people in the world like that do a lot of talking and no doing. And then you get a lot of people who are extremely quiet on meetings, but they are the worker bees. They just get things done. So recognize who is what kind of personality. And the people are the most important. So the goals are very important, but without the people, there are no goals. There's no activity. The people, invest in people and relationships with people. The key to success, hate to say it, money is the key. So there's nothing like money to bring people together. It's hard to get people to volunteer, and we have, it works, but money helps. Delivery, because if you're talking to, if you're getting money from a funder, if they don't see delivery, you might have delays in that, but keep that communication going. Say, we haven't done this, but we've done that. Think about where you can have quick wins that you can report back to show you've got some delivery while you're getting over the hurdles of the other things you should be doing. Flexibility, life changes, techniques change, technologies change, everything changes. So if you set a 5-year plan, that will change because the world changes, and so you need to be able to be flexible and adaptable to that. Think about innovation. What can you do in your network that nobody else does? Don't do the same old stuff. So we did loads of new things in HJ Bionet that I think were really quite innovative, from our node accreditation exercises to pushing the young investigators to be, shadow teams. We had observer teams. Think about how you can be innovative in your network to make an exciting place to be in, and be internationally relevant, because that's how you'll get more funding and that's how you'll get visibility. There are many challenges in running a big network. So there are country-specific issues in terms of, we had Sudan in our network. Huge challenges, obviously, with the war and people trying to work and not being able to get paid. Travel for meetings, just getting a meeting together is really hard because you've got visa issues, you've got outbreaks happening. If there's an Ebola outbreak and you've just spent $20,000 to have a meeting there and nobody can go there. Politics, wars, who's having an election coming up? These are all things we have to think about when we try to have these network meetings. Build capacity in those who need it. So when you see that somebody's not being able to contribute, it could be that they simply don't know how. So what we do is we get them to shadow somebody who does, and then they can build their confidence and their skills, and then they are able to contribute in the future. Keep everyone engaged. It's really hard to do, but if people feel like they're not being reached, then they're going to lose interest in the project. So keep them engaged. Have meetings, but the problem is that there are lots and lots of meetings when you've got a big network. There's always a problem with unequal contributions. The story I told you about before, the talkers and the non-doers. Sometimes those talkers are important because they have good ideas, but then you just need to manage your expectations as to who's going to be doing the work and who's going to be talking about the work. Managing different personalities, the same thing again. Some people are very quiet and don't like to be asked questions. Some people like the spotlight. Let everybody have their personality come out as it needs to, but obviously still managing it that it's, people are not trampled on by the people who like to talk a lot. And then managing national laws, managing, all sorts of university, institutional policies and things. It gets very tedious, the bureaucracy, and then loads of meetings. I spend my life on meetings. Recommendations for running a network: reduce the number of meetings. Hire a good project manager and support staff. There's nothing like a good project manager. They can really take a huge burden off you. Share resources, and then leverage existing resources. So don't start trying to reinvent the wheel. So I've just told you about a whole lot of resources that are already available. Come and use those. They're all open, they're all available, they're all free to use. Don't try to create things from scratch. Get a good project management system. I'll show you how to collab. Set up good communication structures. So some people hate Slack, others only work on Slack. Some people just like mailing lists. So think about how you're going to communicate with different groups. Set up collaborative spaces, so ways for people to share documents, standard processes, agree on policies, etc., and project plans. So this is Active Collab. This is just a project management system, but there are many out there that you can use. If it's a big complex project, or even a small project, it's actually really a good idea just to, even when you're managing your own lab, just to set up some sort of management system so you can keep on track of things, because genomics has many different components. We set up a whole lot of policies. So for a big network like this, you need policies on travel, in this case, we did standards on software development and resource development, so that when you're developing a software for us, you follow certain standards. And then we had roles and responsibilities. We had terms of reference for everything, for all our work packages, for our working groups. Everyone had a set of terms of reference, so you understand why you're there and what you're supposed to achieve. Agree on authorship, always a contentious issue in a network or any collaboration. Who's first author, who's last author, who gets on the paper, who shouldn't be on the paper. Plan this up front so that the expectations are managed. We had what's called the manuscript concept document, where you could fill in all the information so that before you start the project, you've agreed. Obviously, it might change a bit depending on who ends up writing the paper, who ends up jumping out as doing the most work, but at least you have something in there so that it's not a surprise at the end. And we always try to give the junior academics opportunities. I don't need any more publications, I don't care. So I'd rather have the junior academics be the lead authors on publications, because they're the ones that are building their careers. And then finally, sustaining collaborations. The hardest thing in the world. Every grant I've had to write in the last couple of years has said, put a sustainability plan in. If I knew how to sustain this, I wouldn't be applying for money. I think it's the dumbest thing to put in to make you right, because nobody in the world has solved the sustainability thing. We have whole organizations just trying to work out how to do sustainability for example, global biodata resources. But try and think about it. Maybe there's some way you can make some income off the stuff that you do. Maybe you've already got some other grant proposals sort of in line to sustain people. Maybe you can take use this as preliminary results for a new plan, a new project. Think about innovative ideas that can spin off of this. Have these ideas ready so that when a call comes up, and your partner's ready, so that when a call comes up, you're ready to jump in and respond to a new call. Sustainability of resources is also quite difficult. So if you've got new sequencing machines, somebody's got to maintain them, the service agreements and the people. Same with computing resources. It's really difficult to sustain. But think about whether you're going to use them long term and how you might get maybe others in the institution who might want to use them to help sustain the resources that you've developed. Sustaining collaboration. So even though Eshrei Bionet is finished, we still have people from Eshrei Bionet who joined 12 years ago volunteering to work on our projects. So we still work together because we like each other. We invested in these human partnerships, and people just want to contribute. They want to be part of something. So it's really important to invest in the human partnerships. I always remember when I think about some of my peers, I know there's certain people who, when I talk to the others, they say, no, I'll never work with that person. People remember if you did something to annoy them. So it's really important to respect people, even if you have to be the bigger person and say, okay, you were right, because in 2 years' time, when they're looking for a partner for a grant proposal, they will remember if you didn't deliver or if you annoyed them. People remember. So be the bigger person sometimes when it might come down to that, because it's you're investing in your own future, in your own networking future. And when you're trying to do networking, meet people face to face. Don't be shy to go up to them, because when I see something on an email, if I've seen their face, I'm far more likely to remember and to be keen to work with somebody. So with that, I'd just like to acknowledge all the people that are on these different programs that I talked about, and everybody from all the networks that I've been involved in. I mean, HJ Binders had 500 people in the network. And so everyone had a contribution, and I'd like to recognize them. So thank you.